So we are really delighted to have Dr. Erin Crowgey from Nemours to give us a talk on analyzing genomics data. But before I introduce her, let me mention some housekeeping issues. So as you know, all the instructions to obtain CME credit are on the screen. And we also can email you the instructions if you provide your address on the sign-in sheet. The next ID series, so tomorrow we don't have an ID series, that will be next week. We have Dr. Karen Tolson from Christiana Care. We'll talk about opioid overdose education with naloxone take home in the emergency department, so you don't want to miss that one. And please register through the Christiana Care portal, and the full ID series is scheduled. The full ID series schedule is posted on the ACCEL DE-CTR.org. And our November 14th Tech Talk is from Dr. Lewis Frey, one of our collaborators from Medical University of South Carolina. And the title will be forthcoming. Okay, so I'm really very excited to introduce Erin. Dr. Crowgey is the Director of Computational Medicine at Nemours Alfred DuPont Hospital for Children, and she's also the chair of the Genomics and Translational Bioinformatics Working Group within the American Medical Informatics Association. She obtained a PhD in bioinformatics and systems biology from University of Delaware. She already has a large number of publications, although she's very young, and she is PI on a very large Moseley Foundation grant, which goal is to develop a comprehensive oncology genomic program from Nemours, as well as an UNDA quantum grant to develop and implement error-corrected sequencing and single-cell sequencing techniques for studying clonal diversity in pediatric cancers. So our research focuses on establishing comprehensive computational pipelines that will enable the use of omics data in clinical research projects, with the ultimate goal of applying these methods in real time at the bedside. So thank you very much, Erin, for being here, and we look forward to your talk, your presentation.
Oh, thank you so much for that introduction. It's a pleasure to be here. I'm also part of the CTR, and I have the privilege for working in Claudine's core, the bird core, and with Stuart from the University of Delaware. So today I'm going to talk about some data analysis techniques that we have been developing for analyzing genomics data. So real quick, I just wanted to introduce the team. So we truly are a multidisciplinary team, which I'm extremely proud of the way that we've been able to develop our structure. So we have bioinformaticians, so myself, Carl Franke, who's a postdoc, Jeff Myers, who's floating above the next seat here. He wasn't there for the picture day, so we put him above there. I promise his body is not a machine. And Jeff just finished the certificate program with Kathy Liu in bioinformatics as well. We have web bench scientists. That's Alan Robbins. He does all of our library preps and sort of prepares all of the DNA and RNA for our sequencing and does all of our molecular biology assays. And we currently have a position posted for another molecular biologist. So if you know of somebody looking for a job, send them my way. We have application developers, Derek and Scott. They work a lot with the Delaware CTR. Derek is actually our lead web application developer for the Delaware CTR here in the state of Delaware. We work extensively with collaborators, a lot of clinicians, obviously, from Nemours. So we support a lot of MDs and MD PhDs with their clinical research efforts at Nemours. And we do work with external collaborators too, but we're very much really focused right now on our Nemours collaborators and supporting them. We work heavily with University of Delaware, many different PIs in our Delaware institutes as well, MUSD, Delaware State University, and Christiana Care. I have several grants with Todd Julie at the Washington University School of Medicine. That's who I work with through the Children's Oncology Group to get access to the samples that we have and to develop these techniques. I just want to introduce the team because throughout this talk I'm going to use the word we quite often, and this is the we that I'm applying to. My grandmother used to say when I said we, do you have a mouse in your pocket? So I promise I do not have a mouse in my pocket today. The we will be this amazing group of people here that we all work together. So our focus is really, our primary focus, especially for me as a PI, is pediatric cancer research. That's where I've invested a large deal of time in the last 4 years, specifically in omics. So genomics, transcriptomics, sort of all the omics flavors that you can think of, and sort of developing translational bioinformatics techniques that are appropriate for studying pediatric cancers. We have a very large focus as well on molecular biology, and that's because we like to make custom NGS assays. We often find what's commercially available off the shelf that you buy a kit. They usually don't work well for us. It's very difficult just to go in and buy something commercially made and just plug and play and implement it for pediatric cancers. We found that that actually was not working too well, and I'm going to explain that throughout this talk. We love data analytics and big data. I'm going to show you a big data life cycle that was actually presented at NERIC. That is sort of how we approach these types of projects. And there's a lot of machine learning in the later part of this talk, and how we're applying machine learning techniques to genomics data. We do work on application development and core collaborative projects. I'm not going to actually talk about these, though, today. It's not the focus of this particular talk. So it's an exciting time, right? So in the last 10 years, we've seen just huge advancements in technologies, especially as it relates to next generation sequencing or sequencing DNA molecules, and also mass spectrometry. In parallel, the imaging world and all that has also taken off, although we have not focused too much on that. But it's a very exciting time because all of these new technologies, especially in the omics era, are driving new approaches. And it's really opened up a unique niche, because you couldn't have gone to school to learn about these techniques and how to analyze this data because it didn't exist until last year, right? So it's a really exciting time, and it's also a challenging time because we're not training the next generation effectively yet to be able to handle this type of data. And I'm actually going to highlight some stuff that Kathy Wu is doing at University of Delaware to help us with this limitation. Data integration is becoming more and more essential. We've gotten really good at processing large data. We have not been as good or successful at integrating all of those large data sets across institutions, across different types of data elements. So for us, that's phenotype data or electronic healthcare record data, and trying to integrate that with all of this omics data. There's still a lack there in terms of data integration. We've gotten really good at processing the data and figuring out how to actually generate meaningful results. However, we are still struggling in understanding how to effectively integrate this and make it scalable. I call it connecting the dots. Applied translational science, so collectively then, this is really, in my opinion, driving new precision medicine efforts. And we really are centered on driving patient-powered research, right? So we really want the patient to be in the heart of the research that we're doing. We want to be able to hopefully, in the future, deliver personalized treatments based on an individual's genomic or genetic makeup. And so being able, again, to integrate all of this and to effectively analyze this type of data is becoming essential in order to drive these new sort of precision medicine initiatives. So our approach, and I will stand by this forever and ever, good data, right? So there was a paradigm, especially a couple years ago, and certainly if you followed the MD Anderson and Watson situation, you've probably heard of this. Machine learning and data analytics does not fix bad data. Bad data is bad data. Garbage in is garbage out. So the theory that, machine learning and advanced analytics can fix bad data or garbage and turn it into clean dishes is not withstanding the test of time, right? It's just not working out that way. We've seen that with electronic healthcare record projects because that data is technically inaccurate, it's not pristine, it's not curated. We've certainly seen that with omics data as well. So what we really try to focus on is having good data, which means you have good data science techniques. This really requires, again, bringing across domain experts, math and statistics, and computing skills. So at Nemours, we're really lucky that we have both a research center and our clinical, we are a hospital, we're a pediatric hospital. So we have access to, and we can assemble these types of teams between our clinicians, our nurse practitioners, our advanced nurse practitioners, plus all the clinical staff required that really have the domain expert, right? I don't have the domain expert for actually applying these treatments at the bedside for pediatric cancers. That's not my area of expertise. But I have to be able to understand that, otherwise we're just developing algorithms in a box and it's never going to have an impact, right? So it's really essential throughout the entire course of a project, in my opinion, that you always are heavily connected to the domain experts. What I think happens, unfortunately, sometimes is we meet with domain experts and they give us an idea or concept and say, go work on it. and we come back a year later and we present something and we're so far off from anything that would make sense for them in terms of application. So this is really essential. You cannot underestimate your biostatistician, your mathematicians, and your statisticians, and they are different than the people that have computing skills. Now, some people are lucky and have both of those skill sets, right? I kind of feel like I kind of go back and forth between these two sort of areas of expertise, and that's really when you combine them is where you start getting effective machine learning. When you start combining domain experts and these math, you get your quantitative research, right? And hopefully over time between the domain experts and your computing skills, so people developing those applications, you're actually developing meaningful applications that are going to have an impact. Not just something that, oh, this is great, I can do this on Google, but is it really going to change the disease course of a 5-year-old with cancer? Probably not, right? And when we bring all of this together, we hope to deliver a high-quality data science infrastructure that will focus on delivering high-quality data. This came from Ohio State University, who are sort of leaders in this area. All the references are at the bottom of my slides. So this is a concept that we've really embraced. I first saw this presented in a forum event. I've modified it quite a bit by Jennifer Whipples from University of Delaware. We were at the NERC conference in Bar Harbor a few years ago, and her and I actually were roommates. We're really good friends, and we sort of worked on this concept, and since then I have modified it. And Jennifer Whipples now actually teaches a course at University of Delaware trying to teach new students, our new next generation of kids, on how to do this type of work. So the first part of a data lifecycle for big data, which is genomics and the type of data I work for, is the easiest part, right? And it's a new paradigm going to data-driven hypotheses, not having a hypothesis and asking it of your data. We have finally embraced this. This is tried and true. Kathy Wu publishes on this and has done a great job at sort of getting that concept out there. So you have big data, you use bioinformatics or informatics, mathematics. You could, maybe I should put quotes around bio here. Any type of informatics would do. And you come up with a hypothesis of the big data. We didn't come in with a hypothesis with the big data. We actually analyze the big data, see how things cluster. Maybe we're doing PCAs, maybe we're looking at linear discriminant analysis, those types of things. From that, then, we generate a hypothesis. The hypothesis for us here, and again, you could substitute this out with anything. This could be a manual chart review if you're talking about electronic healthcare record data. For me, today, the focus is molecular biology. We then take from that NGS data, we come up with a hypothesis. We use different styles of molecular biology techniques like PCR, things that are really standard. We test that hypothesis. That generates very little data, right? NGS to Sanger, very little data. However, the back end of this life cycle is the hardest. How do you then take that little data and turn it into actionable knowledge? This is where most people fall short, right? We have gotten much better in the last 5 years at doing this. And we finally are getting people to accept this concept of data-driven hypothesis. However, we have not successfully figured out how do we then turn this into knowledge that's actionable at the bedside. And I think that's really key and important to keep this big picture in mind when you're working through these types of projects. So I'm going to go ahead and jump into some of the work that we're doing in pediatric cancer research in terms of data analytics for genomics data. So real quick, why is research needed? Relapse is still a significant problem. As you can see here, unfortunately, still about 50% of children with AML still relapse and do not survive. That's way too high, in my opinion, for 2019. Some of the other cancers have gotten better, but several of them are still coming in around the 50% survival rate. Adverse events and toxicity. So unfortunately, children typically receive the same type of reagents that adults do. They're very toxic on kids. So when you're 5 and you're receiving these types of chemotherapies and radiation, by the time you're 18, you're probably going to have a great deal of toxicity and side effects, right? Because you just were exposed to so much as a young child, and this is still very much a significant problem. So if you are lucky enough to survive, you probably are going to have some really bad adverse events due to the harsh treatments that you received. We now know, and I'll show you a couple data slides on this, that pediatric leukemias are different compared to adults. So the genomic landscape between a child that has AML is completely different than a genomic landscape of an adult that has AML. However, we're still trying to treat children with adult drugs. It doesn't work because they have cancer for a different reason, even though we are classified into the same large category. Targeted therapeutic development then is obviously needed, and it's even more needed in pediatrics because the return on investment for our pharmaceutical companies is not as high, right? if I'm doing lung cancer, a lot of people, a lot of adults get lung cancer. You're going to, there's a lot of return of investment and so forth. So we need to really figure out how to help drive better therapeutics in a space where people aren't necessarily going to make a ton of money on this because these are such rare disorders. So I'm going to focus a bit on pediatric AML. So the first thing that's pretty noticeable about pediatric cancers is that there's a tremendous amount of heterogeneity within the somatic landscape. You can see here even like just the breaking up of this, there's not even a single really dominant, karyotype or these rearrangements, with the MML rearrangements coming in the highest with roughly about 18%. So even within AML, in pediatric AML, the landscape of the diversity of these genomic mutations is significant. So this is really important in terms of creating these types of assays and looking to be able to effectively do this, is you need techniques that can handle all of that heterogeneity within your population. The other interesting aspect of this is that about 20% of these children actually have normal. They can't actually find anything using standard karyotyping technique. However, when we apply more sensitive methods such as NGS, we actually are starting to be able to describe the space much better. And today I'm going to focus a bit on the FLT3 internal tandem duplication, which is an important oncogenic event that's actually associated with a therapy. The other interesting aspect of pediatric cancers, and this was published by Foundation Medicine at ASH a couple years ago, is that, and this is on the, here we're going to increasing in age groups, so this is less than 2 to 10, 10 to 20, so forth, up to over than 70. The fusion is the blue, and the single nucleotide polymorphisms, or single nucleotide variants, are in green. And basically, you can see here, right, children are characterized by these much more complex fusions. They're more prevalent, but significantly decreased to not very common at all found in adults, whereas adults have significantly more single nucleotide polymorphisms. And that kind of makes sense, right? So over time, we're exposed, I'm 60, I smoked 3 packs of cigarettes a day. I exposed myself to harsh things, UV light. Those only typically cause single nucleotide changes, right? Children, however, they're born with some very crazy, large chromosomal events happening already. It's not like they didn't wear sunscreen or they've exposed themselves to all these harsh chemicals. They actually have a much different landscape within their genomic mutations in this space. So the goals for our genomic applications were, one, then, to be able to detect structural variants. And this is very hard to do in NGS. That's probably why these were largely missed throughout the last 10 years, is because we were applying adult analytics, adult algorithms to data generated from children. So the adult algorithms are focused on finding single nucleotide variants. That does you no good when you're analyzing pediatric cancer data, because that's not really what characterized their genome. So we knew that we really had to enhance these types of assays that were being developed, and we couldn't just leverage, what worked in adults, because that's not going to work in kids. We did, though, want to be able to have single nucleotide variant detection, and that's mostly related to the germline landscape. We do know that germline mutations in combination with your somatic landscape are really important, right? There's this cancer predisposition hits. There's things that actually allow these micro environments to allow these tumor types of cells to thrive in. We all have cancer, right? We just keep it in check. I have cancer cells running around my body right now. My body just keeps them in check. They don't thrive, and that's probably because I have a pretty healthy germline landscape that allows that. So why would somebody get cancer on the first day of life, right? We do think the germline landscape is important, so we did want to be able to do that. Ultimately, though, as I said in the beginning, relapse is still a significant problem, right? We need to be able to monitor these types of genomic lesions throughout the entire course of disease, and this is called residual minimal disease monitoring, and this is important because of a limit of detection. And when we first started this project, our limit of detection requirements actually were below the error rate. So the error rate was too high for us to be able actually to deliver appropriate limit of detections, and I'm going to talk to you about how we were able to overcome that. It's of no surprise, and I just had to throw this up here, you can't give a genomics talk without having the wonderful slide that everybody shows, where the cost of sequencing have actually outcompeted Moore's law, which nobody really thought was going to happen. And we are now at, goes up to 2017, We are at $1,000 now to sequence your entire genome, which is impressive. Now, I will say that's the cost for sequencing. You might as well triple that in terms of cost of data analysis. So we've done a great job with decreasing the cost for sequencing. However, we have not done a great job with decreasing the computational cost for analyzing that data. And I'm going to talk a little bit about what we've done to help in that space as well. So high level, right? So I've been rambling about NGS and genomics and so forth. High level, what does that mean? Well, your first step is you have a library prep, right? So you either start with DNA or you start with the RNA, and you have to capture the molecules of interest. There's a million different flavors to do this. You can do this in terms of whole exome, where you're capturing just the exons, the coding regions within a genome. You could do the whole genome. You could use anchored multiplex PCR or an amplicon-based approach. For RNA, the same is true. So if you're doing RNA, you can actually do a poly-A capture. So you're capturing all of the RNA molecules that have a poly-A tail. You could just do a ribosomal depletion, and then therefore you're going to sequence all the different RNA molecules, not just the ones with poly-A tail, right? So this is really important, and again, this is where I see most people make their mistakes. They don't match science and technology to their question and the biology of interest. So if I'm interested in studying coding variants, then I should be focusing on whole exome and not whole genome sequence. Right? If I'm interested in doing a differential gene expression analysis, I better pick a poly-A tail capture technique versus a ribosomal RNA depletion, because it's not going to get me where I need to go. And it's very complex, actually, to figure this out and then ultimately to match it to the bioinformatics. The reason why I skipped a little bit quickly over the sequencing platform is because there are a ton of different sequencing platforms out there. It should come as no surprise, though, that Illumina is sort of the dominant powerhouse in this space, and that's what we use. So we have Illumina instruments at Nemours. We have a NextSeq and a MySeq, and we also work with a lot of sequencing centers, but they all focus on Illumina. I think it's important that people know that there's different types of NGS instruments out there, but for today's talk and for the future of the type of work that we do, right now we are very much focused on the Illumina sequencing platform. So I'm not going to spend too much time on going over this aspect today. In terms of bioinformatics, again, this is where the complications come, right? So how do I know which algorithm is appropriate for me to use on my data set? I'm going to show you a fun slide about how we have to figure that out. So library preparation and sequencing. So high level, you have different ways for capturing your molecule. That's the first thing that you start with, right? So you have to select a capture technique that's appropriate for the biology and the hypothesis that you're asking. Once you capture this molecule, the rest of this is identical. It doesn't really matter, right? So you have your fragment, however you captured this fragment, and you have these different, obviously, millions and millions of these fragments will be generated. And then we do sequencing. When we sequence, we actually only sequence the 5' to 3' end, and then coming into 3' to the 5' end. And the middle here is actually not sequenced, right? But that's pretty important in terms of data analysis, because once I know what my fragment length should be here, when I map this to a reference, depending on how far apart these are mapping, tells me how complex is the arrangement in that region, right? So if I know that this is 300 base pairs, then these should be mapping exactly 300 base pairs apart. When I map them to a reference genome and that doesn't happen, that tells me that individual has a significant rearrangement compared to the reference genome that we're using to do the data analysis. So it's really important to understand this concept, but the hardest part really is trying to figure out, how do I capture the right molecule? If you don't capture the molecule, I don't care how good you are at math statistics, you're not going to write an algorithm that detects it because you never captured it to begin with. So again, a really important concept.
Mm-hmm. So I'm sorry for my ignorance here, but why don't we sequence the middle part?
Yeah, because you could if you wanted to. But you start to, it really complicates the sequencing. So we really want these fragments to be large, but Illumina is short sequencing. So once you get over about 150, 250 base pairs, the quality starts dropping off. So if I wanted to sequence this entire molecule in one foul swoop, all 300 base pairs, the quality of the sequencing towards the later end would be so bad that I would not be able to use it. So it's really about the technology. Illumina is short read sequencing. It's really, really good at generating sequence data between 75 to about 150 base pairs. Their new chemistry goes all the way up to 300. We have not seen a lot of high-quality data coming off of the 300 flow cells yet for the assays that we're running. And if we limited our fragment size to that small size, then we wouldn't be able to find the complex structural variants, because there's not enough space to get that mapping to show that it's not mapping appropriately. Does that answer your question? So, and this is probably one of the next most common questions I get, which is what's the bioinformatics pipeline is best for me, right? So bioinformatics analysis of NGS data can be divided into three major sort of categories. So first you have your wet bench technique, which I talked about, right? So how do you capture those molecules? Are you using hybridization, an amplicon-based approach, amplicon with error-corrected sequencing? Once you generate that data, however, no matter what type of wet bench technique you use, this is pretty consistent in terms of the bioinformatics data processing. The first step is genome alignment. The first big step is genome alignment. There are a ton of algorithms for doing genome alignment. Some use forced reference alignment, so that means we have a reference, we force our reads to map to that reference, and then we call variants based on that. Other approaches use de novo assembly, so we don't actually have a reference, we're actually just throwing all the reads together and allowing them to assemble and build large contexts. So there's a ton of different flavors for genome alignment. Variant detection as well, thousands of algorithms for doing variant detection, and variant annotation. How do I then annotate all these variants that I found? Again, hundreds of applications to do that. There is no gold standard right now for doing this type of work. And the unfortunate aspect of all of this, or the fun aspect for me, is that chances are you need all of these algorithms for your project, right? So in the beginning, I said children have structural variants, adults don't. We have to have a pipeline that can do structural variants. I told you I'm interested in single nucleotide variants because of germline mutations, and some of those are still significant in kids. That's a different algorithm for actually finding those. We're interested in indels. Depending on the size of your indel, GATK works great for less than 25 base pairs. If you're going over 25 base pairs, you better use something like Pindel. So at the end of the day, the answer is you probably need a lot. And you need all these different paths, and you're going to have to get really good at data integration to be able to bring all of those process results back together again. So that's what we set out to do. We set out to create a very comprehensive way of capturing these molecules, and then we set out to figure out how to set up a very comprehensive bioinformatics pipeline that can then analyze all of that data. So what we ended up on doing is we chose a technique that allowed us to use either blood heel sticks, so we can actually use heel sticks, saliva or spit. You actually don't need a lot of input because of the way we're capturing the molecules, and you can use standard isolation techniques. So that's sort of what we decided on, that our capture technique would allow for this to be sort of the starting input. In terms of how we're then capturing the molecules after you extract your nucleic acid, we have several different new techniques we've developed. I'm only going to focus on one today due to time. We decided to focus on, for this particular work, it was called anchored multiplex PCR, or otherwise known as AMP. AMP is really kind of interesting in the sense that for a long time, people couldn't use this technique because of the error rate. And we've been able to overcome that error rate, and I'm going to talk about error-corrected sequencing and how we've done that. You only need one specific primer. So we only actually lay down, these are targeted panels, so we know what genes we're after, but we only have to lay down one PCR primer. We're not laying down two primers, right? So if you think about C gene and how we figured this out for crop genetics, it's a very similar concept. When you transgenically modify a crop, you have no idea where your gene went. And if you only have two primers that sit within your gene, you're not figuring out where it landed in the genome because you're just amplifying what you inserted. So we thought about this and we're like, oh, that concept would work really well when you don't actually know what rearrangements you're looking for. We don't actually know the size of these large structural variants, and they vary. We don't actually know if they have a copy number variation, deletion, or insertion or deletion. So we really wanted a very small piece, a very small oligo that was laying down to amplify or capture the regions of interest. So that's why we went with anchored multiplex PCR. It is fast and easy, and you can target many different classes of variants using this technique. There are other types of techniques, such as hybridization, that work extremely well for sensitivity and specificity for the smaller variants, but it falls short for the larger types of variants that we were looking for. The data analysis, we leverage both a high-performance cluster and the cloud. It's very robust and scalable. We integrate now multiple algorithms and all sorts of data from these types of pipelines. Our data analytics now use error-corrected sequencing, which I'm going to explain that in a second. And then we're starting to develop now our downstream machine learning and advanced analytics of all of this finally processed variant data. assay uses error-corrected sequencing. This is a technique that Todd Dooley developed several years ago out of Washington University. And we've worked with another company who is also developing these called Archer DX, and that's who we use to manufacture these particular kits. We don't manufacture, Nemours does not manufacture anything. We leverage collaborators and so forth to actually manufacture. So when I showed the slide, I was talking about those fragments, right? So the fragments of DNA are the blue, okay? And what we do is we actually tag each one of those molecules in your library prep, right? So every time you capture one of these molecules, we're actually tagging them with what's called a unique molecular index. So I know that this molecule is definitely different than this molecule, which is different than this molecule, which is different than this molecule, because this, which is an 8-mer, combined with the 8-mer of this molecule, is a unique sequence, right? It's not represented anywhere else because we put this tag on every single molecule. So what you can see here is that we have a true variant here, which is the red dot. So let's say that we sequence, and what happens is you get a lot of these molecules with this UMI on it. However, you have to think of the fact that not all of our variants are represented equal, right? So that's called your allele fraction. So when you stack these reads up, what you can see is this is a sequencing error. Right, the yellow are sequencing error. It's random. It's not in the same position. When I line up my UMIs, it's not matching. However, the true variant within your UMIs will match identical, even if it's at a low allele frequency. That's how you overcome the error rate. This is really, in my opinion, the only way you can overcome the error rate of NGS sequencing. We then align those to the genome, so we do a forced reference alignment, and now we can actually call these low allelic variants with really high quality confidence that they are true. So this is using using a gene-specific primer amplification, and the total number of DNA molecules amplified for that particular gene-specific. We look for about 99% uniformity with at least 60x coverage. So how do we apply this then? So here's an example. This is actually a somatic mutation. And this child that was present at time of diagnosis at a very high allele frequency. It's a very well-characterized oncogene, and it's actually a pretty well-characterized variant. What we did is that child unfortunately relapsed, right? So they relapsed and did not survive. But we had a sample right after end of induction therapy. So right after their treatment, we were given a sample to see, hey, was that mutation, that lesion, still present after their treatment? And the answer is yes. However, the noise from normal sequencing is too great to actually find that true variant, right? So this is analyzing the data without using that UMI-based approach that I said, right? I can actually artificially scrub that off and do an analysis and pretend it's not there. And this is what a normal sequencing would look like. However, once we applied that concept of error-corrected sequencing, everything else goes away, and the true variant pops right out. And it's at an allele frequency that's pretty low, much lower than the error rate for Illumina. And unfortunately, we were able to show that this child did, immediately after induction therapy, still had this genomic lesion floating around, right? So now we're trying to understand how do we apply this for minimal residual disease monitoring. Now, just because this variant was still around after an end of induction, we don't know yet without a larger N that means you're always going to relapse because it's there, right? We know that you naturally have these variants floating around. Maybe in a different child, if they had a different germline landscape, this wouldn't have come back up again. So we have a lot of work to do, and this is where machine learning is really going to, I think, advance the field. And just a lot of people think that deep sequencing is the answer. That's probably the number one question I get. Well, you just do deep sequencing. That's the same. It's not the same. Deep sequencing is not the same as error-corrected sequencing. And this is some data that we've generated in my lab to demonstrate that. So what you're looking here, each one of these is just a different sample. Over here is the average number of DNA molecules that you're getting. So you can clearly see between here and here, right, the total number of DNA molecules is significantly less, almost half in this sample. However, the unique molecules, which is the red line, is almost identical. So you can sequence this sample 100 more times, 100 more depth, but you're not increasing the actual number of unique molecules. You're just sequencing the same molecule over and over and over again, and because there's no UMI on it, you can't actually correct for the error rate. So if anything, with deep sequencing, you're actually just inducing more noise into your data than you are actually reducing the noise. So this is why we, again, don't really focus on deep sequencing, because statistically, it's not actually resolving the issues at hand. It's actually increasing the noise that you're generating. So this is sort of a high-level overview then of the bioinformatics tools. So I kind of told you here how we decided on the capture method, what we're using for the capture method. We're using error-corrected sequencing. But then just to drive that point home with all those different lines and which path is correct for me, in the end, we use a ton of different. So we're using 3 different aligners for our reference genome. We're using 2 different aligners for our de novo assembly. And I couldn't even list all the variant detections that we're using here because I would have ran out of space, and we're using lots and lots of different algorithms for them doing the actual variant detection. So validation. So we created these assays, yay. We believe that they're of appropriate quality, they have the right statistical approach. We've combined the wet bench with the informatics. So let's validate this. So the first thing we wanted to do was look at the limit of detection for known variants. It's a pretty good way to start benchmarking your stuff. Start with known stuff. So we took a pretty well-known common cell line, a leukemia cell line, MB411, and had the FLT3 internal tandem duplication. It's about 30 base pairs. And we did a serial dilution, right? So we kept the total genomic DNA constant, but we altered the amount of DNA for this cell line. We also used primary bone marrow samples that had various allele frequencies and sizes that were determined using a standard PCR technique, not an NGS technique. And then we have access to longitudinal samples with a single patient with multiple time points collected, where we know what the true somatic variants are at time of diagnosis and relapse, and we wanted to see if we could actually still detect those at an appropriate limit of detection at end of induction. So on the graph on the left is a serial dilution. So you can see what the allele fraction is here of the 330 base pair insert. So we started with 100%. And over here, you're looking at the number of reads supporting the internal tandem duplication with FLT3 normalized to 10 million read sequence per library. It's a standard way of sort of normalizing the data. And what you can see is that even down to 0.1% allele fraction, which is 0.001 for an AF, we are still actually able to accurately detect this FLT3 internal tandem duplication. This is actually the most sensitive assay there is for FLT3 internal tandem duplication generated thus far. Hopefully this manuscript gets accepted. It is submitted to BMC Medical Genomics. It's accepted with revisions, so I like my revision. And we then applied this technique to actual bone marrow samples from patients, and those are along here. Over here is the allele fractions, and those are the blue lines. And what you can see is then applied in a real clinical setting at very different allele fractions. We're still able to accurately find this particular variant. And the size of the insert, where here the size of the insert did not vary. It was the same sample, just a serial dilution. So we needed to make sure that it works across all different sizes of this particular lesion, although it's in the same region. And in fact, we saw beautiful all the way up to, I think, 90, I think this was a 93, or maybe it's just 90, is the largest one we found so far. So this technique works with an appropriate limit of detection, and not only that, we can capture, no matter what your size of your insert is in FLT3, we can capture that molecule using that AMP technique that I described earlier. We then applied this to longitudinal samples from pediatric subjects, and that's what you're looking at here. I just am showing one individual to kind of keep this simple, as simple as I can. So what you're looking at here in the top left is the allele frequency at time of diagnosis, the allele frequency after end of induction or therapy, and then the allele frequency, because unfortunately this child did relapse and did not survive. And what you can see here is that at the end of induction, although very low allele frequencies, we still found that PTN11 mutation. We still found the FLT3 internal tandem duplication, and we still found the WIT1 single nucleotide polymorphism. And these limit of detections are actually more sensitive than the standard base care, which is flow cytometry. However, again, we're still in the process of understanding what does it mean that you still have these somatic variants present at low allele levels after therapy. What does that mean to a clinician when they intensified your therapy? Would they monitor you more? We don't know yet, right? We're still trying to figure out sort of the biology of this and what does it mean. And this is just a way to further explain the data, and I'm not going to go too far into that. The other interesting aspect to this, because I told you we were interested in germline mutations, is that you do see this 1V where it didn't actually, it almost increased because of the treatment. It didn't actually decrease. And this is a new player that everybody's now all interested about, this EZH2 gene, where this person, and this is a germline event, right, because it didn't respond to treatment. So we know this is not probably a true somatic event, because this is sort of flatlining here between the different, and we know the blast cell count here is pretty low. So, it's not a somatic event. But it's not a full germline event either because of the AF. So it's a mosaic event. Pretty interesting. So now we can actually combine mosaicism, understanding the germline and the mosaicism of the germline, with and how that relates to the somatic mutations. So we can also, so then what we did is we actually then screened 20 samples from Nemours, diagnostic samples, to see, one, can we find new variants that were not known originally at time of diagnosis or relapse? Because we think that, again, those standard techniques weren't finding all the interesting variants because they weren't using our technology. And in fact, again, this is submitted, I don't know what's being cut off, but this has been submitted to BMC Medical Genomics, where we did, in fact, we are able to use this technique to find new, what we believe, new somatic events that have previously gone undiscovered. And this is an example in DCHC7, a pretty well-known oncogene, and in fact, we're seeing a pretty significant problem with the retained intron within this region, within our B-ALL subjects. And here's an example where we were able to find a novel spatin-ABL fusion using this technology in a diagnostic sample for a T-ALL subject. So we're in the process of validating these new variants and sort of linking them to potential outcomes and therapeutics. And that really drives home, again, that major big point in the beginning is data integration, right? It's sort of what keeps me up late at night. It gives me lots of wonderful gray hairs. So now we have to figure out how we're going to, at an individual level, integrate all this omics data, right? So at each individual level, we are generating tons of omics data because I just told you we need like 5 different algorithms with multiple techniques, and you have to be able to integrate that per person. But then you also have to be able to integrate that across the population, and an N of 1 is not much to do anything statistically meaningful with. So you have to then be able to bring this data across the entire population. Well, ultimately, we're trying to link it to drugs and therapeutics, so we have to be able to understand all of that within the space of the cell and how that links then to signaling pathways and how those can be targeted and or if they're already being targeted by therapeutics. Phenotype data is essential. Without good phenotype data, how are they going to be able to actually further process this data in any meaningful way or apply machine learning? And then obviously we do a lot of this work in xenograft models because we can't just test new things and new hypotheses on children. We need a good model system. So now that's really where the state that we're in is like, how are we going to be able to handle bringing all of this together in a meaningful way and drive machine learning? So with that, so I'm about a year into my Moseley grant. So we are, and everything you just saw is what we've been setting up to do. But we are now actively sequencing. We should have all the data by November of this year. We're sequencing 1,000 pediatric subjects. Each subject has 2 to 3 time points collected, diagnosis, end of induction, and if they relapse, we have the relapse sample. And each time point was sequenced via multiple assays because we needed RNA, we needed DNA, we wanted the error-corrected sequencing, but we also wanted the amplicon approach. So at the end of the day, I have about 6,000 NGS libraries across these 1,000 subjects that we are in the process right now of analyzing. It's pretty intense. It keeps me up for sure. And we're trying to figure out how to bring this all together. And ultimately, in parallel, then, we're setting up what I often refer to as our knowledge bases, because I don't know how else to describe it, right? You got to bring all this data together in a meaningful way, so we call them our knowledge bases. And that, to me, is really the data integration. So we're heavily focused on data standards. Scalability is a major issue when you start talking about the extent of what we're doing. And I can't stress enough the importance of the high-quality phenotype data. The genomics data we often describe as a pristine glass of water. If we sequence you with our techniques on chromosome 1, position 5, you have a T. I know you have a T. That's very pristine data. The phenotype data, especially from the electronic healthcare record, we often describe as a muddy glass of water. And when you combine muddy water with clean water, you still get muddy water. And that, in my opinion, is the reason why genomics is still very lacking It's not because we're not figuring out now the genomics techniques and the data analysis and bioinformatics. It's really lack of high-quality phenotype data that's accurate and that was put in place not for liability in coding, but is actually relevant to research and doing the type of work we're doing. And certainly the 2009 Meaningful Health Act that was passed for electronic healthcare records did huge, huge strides to help us. But just like when we sequenced the first human genome, Craig Venter's genome, it didn't solve cancer biology, right? And just like the Meaningful Use Act in 2009, it did not solve all of the issues related to data inaccuracies within the EHR system. So I'm just going to do real quick a little bit then about the data analytics that it takes to kind of pull this off in the different areas that we're sort of sub-focusing in to do this work. So we use high-performance clusters or sometimes the cloud, but the cloud works the same way as a high-performance cluster. So you can just think of it as a high-performance cluster. So basic concept is you have your master or your main node that you log into, typically SSH into it, and this is a scheduler. So this node you log in and you say, I want to run a genome alignment. I need 8 cores to do that. I need 36 hours of wall time, and I need these 5 algorithms to be accessible on that particular node. That's your scheduler. You can use Torque, Slurm, whatever. Doesn't really matter. That then scheduler looks across your computing nodes. For us, we have usually anywhere between 16 and 34 processors per node. And it looks to see, okay, which one of those nodes, or maybe multiple nodes, has the ability to execute that job. And you'll get thrown onto that node, and you'll be given those resources. And the cool thing about high-performance clusters, then, is the ability to parallelize your processing, right? So we have a very talented, very lucky postdoc in the lab, Dr. Carl Franke, who joined us from University of Delaware. He came from Dr. Pam Green's lab at the Delaware Biotech Institute. And he loves this type of work. He's getting ready actually to present this poster at the Mid-Atlantic Bioinformatics Conference up in Philadelphia next week. And what he's doing is trying to figure out is how can we better parallelize this processing to get more efficient at doing it. So I just told you we need to use 500 different algorithms for analyzing NGS data. If it takes me 3 weeks to run all of those algorithms, there's no clinical utility there, right? We have to be able to turn this data around within 3 days. So we really need to be able to figure out how to parallelize all of that processing that's taking place. The highlight from this is that graphical processing units, which should be no surprise GPUs, are really powerful and actually more powerful and faster for parallelizing these types of algorithms. However, a lot of the algorithms that we use are not configured for a GPU environment. So there is still quite a lot of work to do. But Carl is working extensively on how to get better optimization using a CPU environment as well. So machine learning. So we've really focused on increasing in the ability to actually do all of that bioinformatics processing that I mentioned to actually generate your variants. And then after you get all of that wonderful variant data, you've appropriately integrated it, then you get to actually apply some fun machine learning to it. I don't have a lot of machine learning yet for the leukemia project because we haven't I haven't gotten all the data from those 1,000 samples that I mentioned and so forth. However, we do have some examples that we've published in the last year related to CP that I'd like to highlight real quick. So just the background, cerebral palsy is the most common physical disability in childhood. Early diagnosis is challenging. Spastic represents the majority of the cases. It's thought to arise due to a disturbance in the brain. In most cases, this is thought to actually happen during gestation, during the first 24 weeks of life. Just like pediatric cancers, early intervention is desirable, but diagnosis is often delayed for several years. So it's a very similar project to pediatric cancer that we're working on. It's the same type of data, it's genomics data, all that kind of fun stuff. So we set out to, develop a novel sort of assay to be able to do that. We worked with GenPro, which is a startup company out of the University of Delaware with Adam Marsh. We use their library preparation technique, which uses methylation-sensitive restriction enzyme digestion, and we enrich for these CpG islands. And then data analysis, the bioinformatics aspect is a methylation load is calculated per each CpG site, and the number of reads supporting your methylation versus the total number of reads is how you come up with a methylation load. Just like variants, it's not all or nothing. It's not like every cell either has it or doesn't have it. Oftentimes, it's very dynamic. So some methylation sites, It's not all or nothing. It could be 10% methylated, it's like 10% of your cells. It could be 100% methylated. It really just depends. And we use very standard, again, bioinformatic techniques for analyzing those with EDGAR and Lima for this particular project. So we did 32 subjects using this technique, this library technique. We published this last year in BMC Bioinformatics. And we had 16 CP and 16 non-CPR controls. The ellipses here represent a 90% confidence interval, and what you're looking at is an ordinate analysis, which is a non-metric multidimensional scaling for this particular one, similar to a PCA. You don't actually know what the ordinates are, right? You just get beautiful separation. So we fed those methylation sites into this non-metric multidimensional scaling, and we saw that we got actual beautiful separation between the two cohorts, which is kind of phenomenal if you think about it, that your methylation patterns can actually sort of maybe be used to classify you, right? And early in life. So early in life, at time of birth, can we classify you based on those methylation patterns as being more likely to have an issue like CP or less likely? So maybe doctors then will intervene earlier in terms of treatment and starting therapies. So that's what we set out to do. So we used on those CPG sites that were found using EDGAR to begin with. There was about 63,000. We then used an ANOVA. And right here, we plotted the top 200 CPG sites using an ANOVA analysis. And again, what you can start to see is we got beautiful clustering. So there's the dendrogram is cut off up top, but we got clustering on subjects, and we also had clustering based on the feature similarities using a dendrogram approach. We then wanted to see, can we use this data to predict blinded samples, to predict samples that we don't know whether or not they're technically CP or CM. So with this, and this concept was developed by Adam Marsh as part of GenPro, and I can't speak highly enough about Dr. Marsh and the way his mind works. I've learned a lot from him. So what we use is an iterative bootstrap model approach to analyze these CPG combinations. So again, we don't actually believe in single mutation complex dysfunction. We actually believe multiple different regions in the genome are probably disturbed, and trying to convolute those combinations of those is really key to understanding complex phenotypes. So we wanted to be able to look at the combinations of these different CPGs, so we use a linear discriminant analysis under the guidance of a machine learning algorithm. So for each model, a random set between 15 and 40 of the CPG sites were selected from the top 200 from our ANOVA analysis. You randomly select, so we have 32 subjects total. We divided into two, our test and our training. So you randomly select 8 controls, you randomly select 8 CP subjects, and you run the LDA. You keep doing this rinse and repeating and switching who's the 8 and the 8 and the 8 and the 8. And for each time you do that, you run your LD analysis 20 times. So your validation set is not used, right? So we use our training set first, and then we test it on these subjects. If the model does not reach an appropriate accuracy, then it's thrown out. And if it does, then it's ranked as a good model. This was done 6.8 million times, so not many. We're evaluated on all these different CPG models. We used the cloud for this. So then what we did is we generated all of these CPG models. We had less samples that were blinded, that were never seen. So they weren't part of our test or training set. These are our true validation samples. We've never seen the model. We took the LDAs for the top 252 best performing models, and we employed a vote system. And so what you're looking at here is the distribution of those votes, of the LDA output. If you fall, and 5 was centered, if you fall to the left and your voting is here, then you are considered a control. If your distribution for the voting's running through here is to the right, you're considered a CP subject. You're more like a CP subject. If there's a green dot, that means we classified you appropriately. If there's a red dot, that means that is not one that we did not. Our receiving operating curve is down here. Certainly could look better. The green is the actual, blue is theoretical. However, I will say it's more sensitive and accurate than the current diagnosis for CP. So we're actually very pleased with these results, and we are trying to scale this up across more samples and subjects. I think the power of the way this was set up and sort of the voting system, when you actually keep running these and you do these predictions, If we would have only predicted, this subject once, and that happened to be the model that we used here, we would have inappropriately classified them. But by looking at the distribution of those probabilities, you can actually really tighten up the results and the accuracy and the sensitivity. And this is exactly what we're trying to do with the pediatric data set, except for we'd obviously like to be able to predict who's at a higher relapse, who's more likely to relapse. And is there something that we can do to help the clinicians and the clinical team understand that better so they can potentially alter or adjust the treatment? So with that, I'm probably getting really close on time, but I'd like to thank the patients and families from Nemours, the Center for Cancer and Blood Disorders, specifically Dr. Andy Kolb, who's the PI, Karen Robinson, Stephanie Lee, and Dr. Robert Akins at Nemours. That's who we did the CP work with. Adam Marsh, University of Delaware, and the Chief Scientific Officer for GenPro. He actually has the space in this building that I just found out about. And Todd Julie, who's my collaborator at Washington University School of Medicine. So with that, I'm happy to take some questions.
For the LDA, when you chose the top 200, how did you get to the 252?
Yeah, trial and error and just trying different things. It was really just those we thought were the best performing. And in terms of, and we're kind of going back through this now, in terms of like figuring out where the sweet spots are to set those thresholds. A lot of times now what we're doing, and this is thanks to Adam has brought this to my attention, is we actually are simulating a lot of data right now. And we're simulating data and spiking in different sort of situations. So we spike in some situations where everybody has the same CPG site, and it's not really dictated by a bunch of different combinations. And then we simulate data where we know where it is different combinations. So we're trying to get better at really defining how we set some of these criteria. At the end of the day, they're the best performing, and we just wanted to focus on those. And why was 253 not as good performing as 252? I can't actually accurately answer right now.
Okay.
Some of it is a little bit subjective. Yeah.
So that was really spectacular.
Wow. Thank you.
What a journey. So in all this context, how do you handle phenotype data? How do you merge them?
I didn't show too much on that. So the difficulties with the phenotype data is one is getting highly curated phenotype data. So we have a whole system in place now for doing that using smart forms and creating a data dictionary, and that's one aspect. The other aspect then is how in machine learning do you combine genomics data with a very different data type and not ruin all the assumptions of these statistical modeling approaches. And I don't yet have a clear answer on that. I'm working with Sunita at University of Delaware and Adam Marsh to try to figure out how do we combine phenotype data for machine learning with the genomics data, because it's not the same type of data.
Right.
And are we breaking assumptions for random forests and so forth to be able to handle those different types of data elements? So we're still in the early process of the machine learning aspect for that. In terms of just sort of standard data analysis, it's pretty general stuff that we use from the phenotype. Did you survive? Did you not survive? Did you have MRD via standard techniques? Yes or no? So it's very limited. It's like 10 to 20 data elements, and it's not very robust at this point.
Well, that would be fascinating.
I'm really excited. Yeah.
How to merge.
How to merge is really going to be interesting. Yeah.
Absolutely. Any other questions from you? Okay, great. Well,
yeah. Sorry, hopefully it wasn't.
No, it's